Okay, so this is the next webcast on plant structure and function. Today we're going to talk about leaf structure, and then we're also, because it's a related topic, we're also going to talk about transport in plants of water and sugar. So the first thing is just talk about leaves and their basic structure and go through that a little bit. I'm going to, first of all, just show you your diagram and how you can label things correctly on that. So I'll kind of go from top to bottom on this overall. First structure that you have here is the cuticle. We talked about that a little bit Thursday in class. And that's just the waxy covering kind of on the surface, so you can kind of see it here at the top. But then there also would be cuticle down here at the bottom that's on here, so you can see that. It's just a waxy coating on the leaf that helps prevent water loss. If you go inside of that, you're going to have the epidermis. It's labeled F on the top is the upper epidermis, and then E on the bottom is the lower epidermis. Just like in roots and stems, the epidermis is really going to be there kind of as an outer layer of protection. Inside the upper epidermis, you're going to have a layer called the palisade layer, sometimes called the palisade mesophyll. What happens in this region is there is a lot of chloroplasts that are found inside these cells, and so that's where a lot or most of the photosynthesis that takes place is going to be in that leaf. There are going to be some chloroplasts in the epidermis as well, but a lot, that palisade layer is just packed with chloroplasts, so a large amount of photosynthesis is going to occur in those cells. If you go a layer below that, you have C, which is going to be the spongy mesophyll. The spongy mesophyll is, as you can see, loosely scattered cells. So the big thing that it can do is it can hold a lot of the gases. The gases, oxygen that's produced in photosynthesis, as well as the carbon dioxide that's brought into the plant that's going to be used in the photosynthetic processes. The other thing you'll find in the spongy mesophyll or spongy layer is you will find veins running through the plant. So when you're looking at a plant leaf and you see veins running through there, basically those veins are full of xylem and phloem. And on this diagram you can see we have those both labeled. The larger tubes are going to be the xylem that are going to transport water up from the roots and the stem to the leaf so that that water can be used in photosynthesis. And the smaller tubes, H, are going to be the phloem. The phloem is going to transport the sugars down from the leaf, down through the stem, and usually those sugars are stored in the roots. Sometimes they can be stored in fruits or other structures, but the most common place for sugars to be stored would be in those roots. And then working our way down, we already showed the lower epidermis, but one thing you'll notice in the lower epidermis is you have stomata. Stomata are those structures we talked about a little bit Thursday in class. We talked about the stomata and the pores that can open and close. On here we have G, which is really the hole in the stomata, and that is called a stoma. And then also on here, it's not labeled, but you'll notice the guard cells. I'll kind of circle a guard cell. The guard cells are going to be able to regulate the opening and closing of those stomata to allow gases in and also prevent water loss. So I believe that's all the structures on your diagram. This is just another picture of the same thing, so you can see the various structures. Here you have the upper cuticle. Whoops, I hit the wrong button. Here you have the upper cuticle. Let me get a pen to outline that for you guys. I'll use green again. So up here you have the cuticle. Then below that you have the upper epidermis. You've got that palisade layer below that. You've got the spongy layer below that. Both the palisade layer and spongy layer in a area of the leaf called the mesophyll, kind of that middle layer of the leaf, right? Embedded in the spongy layer, you're also going to have veins. So in that, you're going to have xylem and phloem. On this diagram, the yellow are supposed to represent the xylem tubes, and the blue are representing the phloem tubes. Notice on the bottom epidermis, you're going to have these guard cells that are going to surround stoma. Together they make up stomata, and so remember those are the pores they're going to let gases in and out, and also hopefully prevent water loss. We'll talk about that here in a second. So this just shows the guard cells and the stoma. Those are embedded in the bottom epidermal layer of the leaf, so you can see that. And then this kind of shows how they open and close. What happens is when there's a lot of water present, like at night when there's dew and there's moisture and that sort of thing, when there's a lot of water present, the water will go into those stoma, and the stomas, those guard cells, I should say, are arranged in such a way when they fill up with water that kind of causes them to go ahead and open up. On the other hand, when they lose water, so in the heat of the day, when it's really hot or dry conditions, then the stoma are going to, or guard cells are going to lose water, and so that's going to cause those guard cells to close the stoma and hopefully prevent water loss when once again, it's hot and dry and the plant needs to hold on to the water as much as possible. Just to kind of go back over this again a little bit, we talked a little bit about monocots and dicots in previous days, but a couple things related topics here. For the veins, you'll notice, as we've talked about before, I'll use red this time. For the veins in monocots, you're going to have parallel veins in the leaves, and in dicots, you're going to have net venation. At least most of the time. It's not 100% of the time on those, but a lot of the trees we've talked about before are going to be dicots, and so a lot of times when you see a plant, if it's got net venation, chances are it's going to be a dicot, not 100%. And that's kind of the major thing associated with the leaves. So we'll move on. Now we're going to kind of shift a little bit into a related topic, and that's transport in plants. So the first thing we want to talk about is just kind of transport of water. The big thing that moves or transports water is you've got a couple of different factors. You've got two things. First of all, down in the roots, you have a thing called root pressure. So what happens is, if you remember when we talked about roots a little bit, there's going to be active transport by the roots of nutrients like nitrogen and phosphorus and things like that are being actively transported in at the epidermis, and then also actively transported in at the endodermis, which is kind of around that vascular cylinder. When those nutrients, those nitrogen, phosphorus, other dissolved particles are pumped in by active transport into the vascular cylinder, that's going to cause a lot of water to be pulled in by osmosis, and that's actually going to force water up in the plant. So that root pressure will kind of actually help pull water up into the plant. There's a couple different factors that are going on as well. If you look at your notes in the lab manual, there's the process of adhesion going on and the process of cohesion. Adhesion is the idea that the water molecules are going to bind well to the tubes, the xylem tubes within the plant, and so that's going to kind of help hold the water. And then cohesion is the idea of the fact that the water molecules will bind to one another, and the combination of those two forces is going to create what's called capillarity, and that's going to tend to move water up in the plant. And kind of moving along. The other thing that's going on that's very important is you're going to have transpiration that's occurring in the leaves of the plant. So there's evaporation from the leaves. We just talked about this stomata and how water can be lost from them. One of the big things with plants is there is always going to kind of be water evaporating somewhat from the leaves. So there's pull up at the leaves for water, and then there's kind of this pull down in the roots, and those two combinations of things are going to move basically water through the plant from the roots up to the leaves. So that's transpiration. Sugars work in a different way. They're gonna move through phloem tubes. They tend to be transported from the leaves down to the roots, or sometimes to other structures like fruit, that sort of thing. And the basically way that the sugars work is there's going to be active there's a lot of sugars that are going to be made in the leaves. I'll get a color here I'll do blue on this one. But there are sugars that are going to be made up here in the leaves. And what happens is those sugars are actively transported into these structures called sieve tubes. I'll circle that name here for you. The sugars are actively transported into the sieve tubes. And then what happens is you have down in the roots, if that's the place where they're going, you have there's active transport of the sugars out of the vascular cylinder, out of the phloem into the cortex. And so that is going to, the pumping, kind of the pulling of the sugars out at the roots and the pumping in at the leaves is going to move that sugar down the phloem tubes. Okay, you call the upper part where the leaves are, you call that the source of the sugar, and where the sugar comes out down in the roots, that you call that the sink, because it's kind of being pulled out. And so you get that combination that is then going to move sugars along. Sometimes in the spring, when new leaves are made, that sort of thing, the sugars will kind of go in a reverse direction. The sugars are being pumped out, actively transported from the cortex of the root into the sieve tubes and phloem tubes, and then actually being pulled out where the new leaves are being made, or up in the branches where those new structures are being made. So it can work in the opposite direction, but usually it's going from leaves, because that's where sugars are being generated, and down to the roots for storage. This is just a picture of kind of how this process is working. Water also helps that process, because the water is kind of moving in the opposite direction and moving up, and it's actually kind of, some water is pulled in to help kind of the sugars flow along. But once again, up in the leaf, that is considered, the source. Let me get back to this if I can. And let me get a pen here. Red. So once again, up in the leaf, that is the source, and the sugars are being made up there, and they're actually pumped into the phloem tubes, or the sieve tubes, or the sieve tube elements, or the cells basically within the phloem tubes. And the sugars are going to be pumped in there. These companion cells are cells that are associated and really alongside the phloem tubes, and they kind of help keep the phloem tubes alive. They do a lot of their basic cellular processes. So the sugars are pumped into the phloem up at the leaf, and then the idea is the sugars are being actively transported or pumped out down in the root or the sink, and that's kind of creating this movement of sugars at that time. The water, on the other hand, is usually being pulled up in the xylem tubes, so it kind of gets pulled out once it gets down to the roots. Obviously, most of the water is coming in from the soil around the plant, but some of this water that's been pulled down with the phloem gets pulled out back in the xylem tube. It travels up, and then a portion of that will get pulled back into the phloem, and that kind of creates the fluid in the sap, in the sugar. So hopefully that helps. So that's our talk about leaves and also transport in plants. Have a good day.